Humoral and cell-mediated immune responses. The body will mount either a humoral response or a cell-mediated response, depending on the antigen which stimulates the system. Rightward's double arrow humoral immunity produces antibodies in response to toxins, XV venom-free bacteria, and viruses present in the body fluids. Rightward's double arrow, humor, is medieval term for body fluids. Here it refers to the fluid of the blood and the lymph. Rightward's double arrow antibodies to these types of antigens are synthesized by certain lymphocytes and then secreted as soluble proteins which circulate through the body in blood, plasma, and lymph. Rightward double arrow cell-mediated immunity is the response to intracellular bacteria and viruses, fungi, protozoans, worms, transplanted tissues, and cancer cells. Rightward's double arrow depends on the direct action of certain types of T lymphocytes rather than antibodies. In humoral immunity or cell-mediated immunity, there are several general steps involved in the immune responses. 1. Antigen must encounter the B lymphocytes, T lymphocytes, and antigen-presenting cells, APCs, capable of carrying out an adaptive immune response. 2. Naive lymphocytes must recognize epitopes of an antigen by means of antigen-specific receptor molecules on their surface and become activated. 2. Naive lymphocytes must recognize epitopes of an antigen by means of antigen-specific receptor molecules on their surface and become activated. This is known as clonal selection. 3. The large clump of identical B lymphocytes, T4 lymphocytes, and T8 lymphocytes now differentiate into effector cells capable of directing body defenses against the original antigen, resulting in its destruction or neutralization. 4. The B lymphocytes, T4 lymphocytes, and T8 lymphocytes differentiate into long-lived, circulating memory cells. Institute for Advanced Studies, Jodhpur 67. In the humoral response, B cells produce antibodies against extracellular pathogens. Rightward's double arrow. The humoral response occurs when an antigen binds to B cell receptors that are specific for the antigen. Rightward's double arrow. The B cells differentiate into a clone of plasma cells which begin to secrete antibodies that are most effective against pathogens circulating in the blood or lymph. Rightward's double arrow memory cells are also produced and form the basis for secondary immune responses. The selective activation of AB cell results from one of two mechanisms. 1. Dependent antigens, antigens that evoke the cooperative response of macrophages, helper T cells, and B cells. These antigens cannot stimulate antibody production without helper T cell involvement. Most antigens are T-dependent. Memory cells are produced in T-dependent responses. 2. T-independent antigens, antigens that trigger humoral immune responses without macrophages or T cell involvement. These antigens usually are long chains of repeating units, such as polysaccharides or protein subunits often found in bacterial capsules and flagella. B cells are stimulated directly by the antigen, which probably binds simultaneously to several antigen receptors on the B cell surface. The antibody production, humoral response, is usually much weaker than that of T-dependent antigens. No memory cells are generated in T-dependent responses. Whether activated by T-dependent or T-independent antigens, AB cell gives rise to a clone of plasma cells. Rightward's double arrow. Each of these effector cells secretes up to 2,000 antibodies per second into the body fluids for its 4 to 5 day lifespan. Rightward's double arrow. The specific antibodies help eliminate the foreign invader from the body. The antibodies produced during humoral immunity ultimately defend the body through a variety of different means. These include 1. Opsonization. Opsonization, or enhanced attachment, refers to the antibody molecules IgE, IgE and the complement proteins C3b and C4b attaching antigens to phagocytes. This results in much more efficient phagocytosis. 2. Antibody-dependent cellular cytotoxicity, ADCC, by NK cells. NK cells are capable of antibody-dependent cellular cytotoxicity or ADCC. NK cells have receptors on their surface for the Fc portion of IgE. When made against epitopes on foreign, membrane-bound cells, e.g., virus-infected cells and cancer cells, the Fab portions of the antibodies react with the foreign cell. The NK cells then bind to the Fc portion of the antibody. The NK cell then releases pore-forming proteins called porphyrins, proteolytic enzymes called granzymes, and chemokines. Enzymes pass through the pores and activate the enzymes that lead to apoptosis of the infected cell by means of destruction of its structural cytoskeleton proteins and by chromosomal degradation. As a result, the cell breaks into fragments that are subsequently removed by phagocytes. Porphyrins can also sometimes result in cell lysis. When NK cells are carrying out ADCC, they are sometimes also referred to as killer cells. Institute for Advanced Studies, Jodhpur 68. 3. Neutralization of exotoxins. For an exotoxin to cause harm, it must first bind to receptors on a susceptible host cell. Antitoxin antibodies, mainly IgE, are made against protein exotoxins. They combine with the exotoxin molecules before they can interact with host target cells and thus neutralize the toxin. 4. Neutralization of viruses. In order for viruses to infect a cell and replicate, they must first adsorb the receptors on the host cell's plasma membrane. Antibodies made against the epitopes on viral capsids or on viral envelope glycoproteins that function in the attachment of the virus to the host cell receptor prevent viral adsorption. See Fig. Neutralizing antibodies are especially important in preventing viral reinfection. 6. Agglutination of microorganisms. Agglutination is mainly a function of antibodies with multiple reactive Fab sites such as IgM and IgA. The antibodies microorganisms together cause them to agglutinate so they can be filtered out of the lymph and blood and be phagocytosed more effectively by the fixed macrophages of the mononuclear phagocytic reticuloendothelial system. See Fig. In the cell-mediated response, cytotoxic T cells defend against intracellular pathogens. Rightward's double arrow antigen-activated cytotoxic T cells kill cells that are infected by pathogens, e.g., viruses, bacteria. Rightward's double arrow host cells infected by viruses and other pathogens display antigens complex with class 1 MHC molecules on their surfaces. Rightward's double arrow cytotoxic T cells have specific receptors that recognize and bind to antigen class 1 MHC markers. Note that this differs from helper T cells which bind to antigen class 2 MHC complexes. Rightward's double arrow. The cytotoxic T receptor can bind to any cell in the body displaying the antigen class 1 MHC marker since class 1 MHC is present on all nucleated cells. Rightward's double arrow. When a cytotoxic T cell binds to an infected cell, target cell, it releases chemicals that form the lesion in the infected cell's membrane. Rightward's double arrow cytoplasm escapes through the lesion and eventually cell lysis occurs. Rightward's double arrow destruction of the host cell not only removes the site where pathogens can reproduce, but also exposes the pathogens to circulating antibodies from the humoral response. Rightward's double arrow cytotoxic cells continue to live after destroying the infected cell and may kill many others displaying the same antigen class 1 MHC marker. Institute for Advanced Studies, Jodhpur 69. Helper T lymphocytes function in both humoral and cell-mediated immunity. Cells that take up antigens, such as B cells and macrophages, alert the immune system, via helper T cells, of the presence of a foreign antigen. Rightward's double arrow such cells engulf foreign material, e.g. bacteria. Double arrow the foreign protein, e.g. from broken down bacteria, binds to a class 2 MHC molecule and is conveyed to the outside of these cells. Rightward's double arrow the class 2 MHC molecule with bound foreign antigen is recognized by a helper T cell. Helper T cells that bind to a class 2 MHC antigen complex on these cells are induced to differentiate into either of two clones of cells. 1. Active helper T cells secrete chemicals that stimulate other lymphocytes, e.g., stimulates differentiation of B cells into antibody-secreting plasma cells and induces cytotoxic T cells to become active killers. 2. Memory helper T cells. These cells become important when there is a second exposure to the antigen E11. Immunity in health and disease. Immunity can be achieved naturally or artificially. Active immunity is the immunity conferred by recovery from an infectious disease. Rightward's double arrow depends on response by the person's own immune system. Rightward's double arrow may be acquired naturally from an infection to the body or artificially by immunization, also known as vaccination. Rightward's double arrow vaccines may be inactivated bacterial toxins, killed microbes, parts of microbes, or viable but weakened microbes. Rightward's double arrow in all cases the organisms can no longer cause the disease but can act as antigens and stimulate an immune response. Rightward's double arrow a person vaccinated against an infectious agent who encounters the pathogen will show the same rapid, memory-based secondary response as someone who has had the disease. Passive immunity is immunity which has been transferred from one individual to another by the transfer of antibodies. Rightward's double arrow naturally occurs when egg antibodies cross the placenta from a pregnant woman to her fetus. Rightward's double arrow some antibodies, IgA, are transferred to nursing infants, so breast milk. Rightward's double arrow provides temporary protection to newborns whose immune systems are not fully operational at birth. Persists as long as the antibodies last, a few weeks or months, but it can provide protection from infections until a baby's own immune system has matured. B. The immune system's capacity to distinguish self from and self limits blood transfusion and tissue transplantation. The body's immune system distinguishes between self, the body's own cells, and non-self foreign cells. Non-self includes pathogens and cells from other individuals of the same species. 1. Blood groups and blood transfusion. The human ABO blood groups provide a good example of non-self recognition. The antigen present on the surface of the erythrocytes is not antigenic to that person but may be recognized as foreign if placed in the body of another individual. Rightward's double arrow individuals of blood type A have the A antigen and make anti-B antibodies. Institute for Advanced Studies, Jodhpur 70. Rightward's double arrow individuals of blood type B have the B antigen and make anti-A antibodies. Rightward's double arrow individuals of blood type AB have the A and B antigen and make no antibodies. Rightward's double arrow individuals of blood type O have neither the A nor B antigen and make anti-A and anti-B antibodies. Blood group antibodies can cause blood of a different antigenic type to agglutinate, resulting in a life-threatening transfusion reaction. Rightward's double arrow type O individuals are universal donors since their blood has neither antigen. Rightward's double arrow type AB individuals are universal recipients since they produce neither antibody or antibody B. Rightward double arrow the blood group antibodies are present in the body before a transfusion occurs because they form in response to the body's normal bacterial flora that have epitopes very similar to blood group antigens. This phenomenon is termed as cross-reactivity. Usually M-class antibodies do not cross the placenta, thus they present no harm to a developing fetus with a blood type different from the mother. Rightward's double arrow the Rh factor is another blood group antigen. Rh factor causes problems when a mother is Rh negative and her fetus is Rh positive, inherited from the father. Rightward double arrow when small amounts of fetal blood cross the placenta and come into contact with the mother's lymphocytes, the mother develops antibodies against the Rh factor. Rightward double arrow usually only a problem in a second child since the response will be quick due to sensitization and formation of memory cells during the first baby's gestation. Rightward's double arrow unlike blood group antibodies, Rh antibodies are Ig class, which can cross the placenta. Rightward's double arrow the mother's antibodies cross the placenta and destroy the red blood cells of the Rh positive fetus. 2. Tissue grafts and organ transplantation. The MHC is a biochemical fingerprint unique to each individual. Rightward's double arrow complicates tissue grafts and organ transplants since foreign MHC molecules or antigens and cause cytotoxic T cells to mount a cell-mediated response. In the case of bone marrow transplants, which are used to treat leukemia and other cancers, the graft rather than the host is the source of immune rejection. Rightward's double arrow the donated bone marrow contains lymphocytes that will react against the recipient. Rightward's double arrow this graft versus host rejection is limited if the MHC molecules of the donor and recipient are well matched. The reactions of the immune system to transfusions, tissue grafts, and organ transplants are normal reactions of a healthy immune system, not disorders of the system. C. Abnormal immune function can lead to disease. 1. Allergies hypersensitivity. Allergy equals a hypersensitivity of the body's defense system to an environmental antigen called an allergen. Believe these reactions to be evolutionary remnants to infection by parasitic worms due to similarities in the responses. IgE class antibodies are commonly involved in allergic reactions. These antibodies recognize pollen as allergens. Rightward's double arrow IgE antibodies attach by their tails to non-circulating mast cells found in connective tissues. Remember, mast cells are involved in the inflammatory response. Rightward's double arrow when a pollen grain bridges the gap between two adjacent IgE monomers, the mast cell responds with a reaction called degranulation. Rightward's double arrow degranulation involves the release of histamine, another inflammatory agent. Rightward's double arrow histamine causes dilation and increased permeability of small blood vessels, which results in the common symptoms of an allergy. Rightward's double arrow antihistamines are drugs used to treat allergies, since they interfere with the action of histamine. Brightbird's double arrow anaphylactic shock is a life-threatening reaction to injected or ingested antigens. It is the most serious type of acute allergic response. Brightbird's double arrow occurs when mast cell degranulation causes a sudden dilation of peripheral blood vessels and a drastic drop in blood pressure. Brightbird's double arrow death may occur in a few minutes. Brightbird's double arrow. This hypersensitivity may be associated with foods, peanuts, fish, or insect venoms, wasp or bee stings. Brightbird's double arrow epinephrine may be injected to counteract the allergic response. 2. Autoimmune diseases. Autoimmune disease equals an immune system reaction against self. Examples. Brightbird's some cases involve immune reactions against components of the body's own cells, which are released by the normal breakdown of skin and other tissues, especially nucleic acids DNA, RNA, in systemic lupus erythematosus SLE. Brightbird's double arrow rheumatoid arthritis is an autoimmune disease in which inflammation damages cartilage and bones in joints. Brightbird's double arrow destruction of insulin-producing pancreas cells by an autoimmune reaction appears to cause insulin-dependent diabetes. Brightbird's double arrow in multiple sclerosis. T cells reactive against myelin infiltrate the central nervous system and destroy the myelin of neurons. Brightbird's double arrow antibodies produced to repeated streptococcal infections may react with heart tissues and cause valve damage in some people. Brightbird's double arrow other autoimmune diseases are Graves' disease and rheumatic fever. Institute for Advanced Studies, Jodhpur 71. Institute for Advanced Studies, Jodhpur 72. 3. Immunodeficiency diseases. Immunodeficiency refers to a condition where an individual is inherently deficient in either humoral or cell-mediated immune defenses. Severe combined immunodeficiency, SCID, is a congenital disorder in which both the humoral and cell-mediated immune defenses fail to function. Not all cases of immunodeficiency are inborn conditions. It's double arrow. Some cancers, like Hodgkin's disease, damage the lymphatic system and make the individual susceptible to infection. Brightbird's double arrow. Some viral infections cause suppression of the immune system, e.g., AIDS. Acquired immunodeficiency syndrome, AIDS. Acquired immunodeficiency syndrome is a severe immune system disorder caused by infection with a human immunodeficiency virus, HIV. Individuals with AIDS are highly susceptible to opportunistic diseases, infections, and cancers that take advantage of a deficient immune system. Mortality rate approaches 100%. HIV probably evolved from another virus in Central Africa and may have gone unrecognized for many years. There are two major strains, HIV-1 and HIV-2. HIV infects cells, including helper T cells. Brightbird's double arrow after entry. HIV RNA is reverse transcribed and the product DNA is integrated into the host cell genome. Brightbird's double arrow in this provirus form, the viral genome directs the production of new virus particle. Brightbird's double arrow HIV is not eliminated from the body by antibodies for several reasons. Brightbird's double arrow. The latent provirus is invisible to the immune system. Brightbird's double arrow. The virus undergoes rapid mutational changes in antigens during replication which eventually overwhelms the immune system. Brightbird's double arrow. The population of helper T cells eventually declines to the point where cell-mediated immunity collapses. Researchers are not entirely sure why this occurs. Brightbird's double arrow. Secondary infections characteristic of HIV infection develop pneumocystis pneumonia and Kaposi sarcoma. Institute for Advanced Studies, Jodhpur 73. E12. Immunization and vaccines. Immunization is the means of providing specific protection against most common and damaging pathogens. Specific immunity can be acquired either by passive or by active immunization, and both modes of immunization can occur by natural or artificial means. Passive immunity. Immunity can be acquired without the immune system being challenged with an antigen. This is done by transfer of serum or gamma globulins from an immune donor to a non-immune individual. Alternatively, immune cells from an immunized individual may be used to transfer immunity. Passive immunity may be acquired naturally or artificially. Naturally acquired passive immunity. Immunity is transferred from mother to fetus through placental transfer of egg or colostral transfer of egg. Artificially acquired passive immunity. Immunity is often artificially transferred by injection with gamma globulins from other individuals or gamma globulin from an immune animal. Passive immunity with immune globulins or gamma globulins is practiced in numerous acute situations of infections, diphtheria, tetanus, measles, rabies, etc., poisoning, insects, reptiles, botulism, and as a prophylactic measure, hypogammaglobulinemia. Situations, gamma globulins of human origin are preferable, although specific antibodies raised in other species are effective and used in some cases. Poisoning, diphtheria, tetanus, gas gangrene, botulism. One form of immunization has the advantage of providing immediate protection. Heterologous gamma globulins are effective for only a short duration and often result in pathological complications, serum sickness, and anaphylaxis. Active immunity. This refers to immunity produced by the body following exposure to antigens. Naturally acquired active immunity. Exposure to different pathogens leads to subclinical or clinical infections which result in a protective immune response against these pathogens. Artificially acquired active immunity. Immunization may be achieved by administering live or dead pathogens or their components. Vaccines used for active immunization consist of live, attenuated organisms, killed whole organisms, microbial components, or secreted toxins which have been detoxified. Brightbird's first live vaccine was cowpox virus, introduced by Edward Jenner as a vaccine for smallpox. However, variolation, inoculation using pus from a patient with a mild case of smallpox has been in use for over 1,000 years. Brightbird's double arrow. The first live vaccine was cowpox virus, introduced by Edward Jenner as a vaccine for smallpox. However, variolation, inoculation using pus from a patient with a mild case of smallpox has been in use for over 1,000 years. Brightbird's double arrow live vaccines are used against a number of viral infections: polio, Sabin vaccine, measles, mumps, rubella, chickenpox, hepatitis A, yellow fever, etc. The only example of live bacterial vaccine is one against tuberculosis, Mycobacterium bovis, BCG. While live vaccines normally produce only self-limiting non-clinical infections and subsequent immunity, they carry a serious risk of causing overt disease in immunocompromised individuals. Double arrow killed heat, chemical or UV irradiation. Viral vaccines include those for polio, Salk vaccine, influenza, rabies, etc. Most bacterial vaccines are killed organisms: typhoid, cholera, plague, pertussis, etc. Brightbird's double arrow. Other bacterial vaccines utilize their cell wall components: Haemophilus, pertussis, meningococcus, pneumococcus, etc. Some viral vaccines, hepatitis B, rabies, etc., consist of antigenic proteins cloned into a suitable vector, e.g., yeast. When the pathogenic mechanism of an agent involves a toxin, a modified form of the toxin toxoid is used as a vaccine, e.g., diphtheria, tetanus, cholera. These subunit vaccines are designed to reduce the toxicity problem. Each type of vaccine has its own advantages and disadvantages. Rightward's double-arrow anti-idiotype antibodies are also under trial. Similarly, DNA vaccines and immunodominant peptides recognized by the MHC molecules are under investigation, particularly for protection against viral diseases. Right-arrow, the protective immunity conferred by a vaccine may be lifelong: measles, mumps, rubella, smallpox, tuberculosis, yellow fever, etc., or may last as little as 6 months. Cholera. Application of immunological techniques. 1. Monoclonal antibodies with virtually any desired specificity can be readily prepared. 2. Proteins can be detected and quantitatively determined by using an enzyme-linked immunosorbent assay, ELISA. 3. Western blotting permits the detection of proteins separated by gel electrophoresis. 4. Immunofluorescent markers make possible the visualization of proteins in the cell using fluorescence microscopy. 5. Gold heavy metal tagged antibodies make possible the visualization of proteins in the cell using electron microscopy. Institute for Advanced Studies, JODPER 74. E13. Immunity against infectious pathogens. Introduction. During evolution, an extremely complex system of anti-infections defenses has emerged. At the same time as vertebrates and mammals developed their defenses, microbes continued evolving as well, and many became adept at avoiding the consequences of the anti-infections defense mechanisms. The interplay between host defenses, microbial virulence, and microbial evasion mechanisms determines the outcome of the constant encounters between humans and pathogenic organisms. 2. Non-specific anti-infectious defense mechanisms. Specific defense mechanisms play a most important role as a first line of defense, preventing penetration of microorganisms beyond the outer exposed surfaces of the body. The following is a brief description of the most important non-specific defense mechanisms. Physical and chemical barriers, including the integrity of the epithelial and mucosal surfaces, the flow of mucosal secretions in the respiratory tract, the acidity of the gastric contents, and the secretion of lysozyme in tears, saliva, and most secretions. The importance of these barriers is apparent from the prevalence of infections when their integrity is compromised. B. Inducible nonspecific responses, including fever and release of interferons, activated when infectious agents manage to invade, particularly effective in preventing viral replication. C. Phagocytosis. As a microbe penetrates beyond the skin or mucosal surface, it will encounter cells able to ingest it. 2 types of cells are particularly adept at non-immune phagocytosis: tissue macrophages and granulocytes, particularly neutrophils. This non-immune phagocytosis involves a variety of recognition systems. Big point 1. 1. CR3 receptors are able to interact with C3b and IC3b on the microbial membrane, generated as a consequence of complement activation by the alternative pathway, a property common to many bacteria. See below. 2. Mannose receptors on phagocytic cells may mediate ingestion of organisms with mannose-rich polysaccharides, such as Candida albicans. Mannose-mediated phagocytosis is amplified by a mannose-binding protein that promotes phagocytosis through complement activation and direct interaction with the C1q colectin receptor on phagocytic cells. 3. C-reactive proteins bacterial polysaccharides and has very similar effects to the mannose-binding protein, activating complement and promoting phagocytosis, both through CR1 and CR3, as well as by other receptors, including the Fc-gamma-RI and the C1q receptor, both of which bind this protein. Deactivation of the complement system via the alternative pathway. A variety of microorganisms, bacteria, fungi, viruses, and parasites can activate complement by the alternative pathway. See Table 1. In most cases where adequate studies have been carried out, polysaccharidic structures have been proven to be responsible for complement activation of the alternative pathway. This activation will lead to phagocytosis, through the generation of C3b, and to chemotaxis, through the release of C3a. Acute phase reaction and leukocyte chemotaxis. The initial recognition by phagocytes and the activation of the complement system by the alternative pathway, by themselves, may not be sufficient to eradicate the invading microorganism, but the response is quickly amplified by a multitude of cytokines released by macrophages activated as a consequence of phagocytosis, including IL-1, IL-8, and TNF-alpha. 1. IL-1 cause an increase in body temperature, mobilize neutrophils from the bone marrow, and upregulate the synthesis of a variety of proteins known as acute phase reactants, including C-reactive protein and the mannose-binding protein mentioned above. 2. TNF and IL-1 upregulate the expression of cell adhesion molecules in the endothelial cells of neighboring endothelial cells, thus promoting adherence of leukocytes and increase vascular permeability. Both. Institute for Advanced Studies, JODPER 75. Factors facilitate the migration of leukocytes off the vessels, toward the focus of infection. 3. IL-8 has chemotactic properties. Together with other chemotactants, such as C5a and bacterial peptides, it attracts neutrophils toward the focus of infection. F natural killer NK cells are able to destroy viral infected cells as a consequence of the delivery of an activating signal in the absence of an inhibitory signal. 1. The activated signal is delivered as a consequence of the interaction between a recognition molecule, NKRP1, on the NK cells and glycoproteins expressed on the membrane of the infected cells. 2. The inhibiting signal is usually delivered as a consequence of the interaction between a membrane protein in the NK cell membrane and MHCI molecules on the membrane of normal cells. Infected cells often have a reduced expression of MHCI molecules due either to a downregulation of cellular protein synthesis or a specific inhibition of MHCI transport to the cell membrane, discussed later in this chapter. GDT lists are predominantly localized to the mucosal epithelia, where they appear to recognize infected epithelial cells by non-immunological mechanisms, i.e., not involving the T cell receptors, which are subsequently immunization. It was eventually demonstrated that the cell wall polysaccharides of several strains of Enterobacteriaceae and the AB oligosaccharides of human erythrocytes are structurally similar. Thus, cross-reactive antibodies to Enterobacteriaceae are responsible for the spontaneous development of antibodies to human red cell antigens in chickens. 3. Newborn babies of blood groups A, B, or O do not have either NA or NAB isohemagglutinins, but will develop them during the first months of life, as they get exposed to common bacteria with polysaccharide capsules. However, newborns are tolerant to their own blood group substance, so they will only make antibodies against the blood group substance that they do not express. Blood group AB individuals never produce AB isoagglutinins. B. Other mechanisms for the generation of natural antibodies. Cross-reaction is probably the most explanation for the emergence of natural antibodies, but other mechanisms, such as the mutagenic effects of TIN-dependent antigens and the nonspecific stimulatory effects of lymphokines released by antigen-stimulated T lymphocytes, which could activate B cells responding to other antigens, could explain the rise of nonspecific immunoglobulins that is observed in the early stages of the humoral response to many different antigens. It is a matter of random probability that some of those nonspecific immunoglobulins may play the role of natural antibody relative to an unrelated antigen. C. The significance of natural antibodies. Natural antibodies may play an important protective role. Antibodies elicited to E. coli K100 cross-react with the polyribophosphate of Haemophilus influenzae and can protect experimental animals against infection with the latter organism. It is logical to assume that such cross-immunizations may be rather common and play an important protective role against a variety of infectious agents. IV. The protective role of specific antibodies. A. The humoral immune response. Antigen not eliminated by non-immunological means and continues to replicate, it will eventually spread through the blood and lymph, and will usually be trapped by macrophages and follicular interdigitating cells in the lymph nodes and spleen. Those cells are able to internalize antigens interacting with receptors such as the mannose receptor, see above, process the antigen in the endosomic compartment, and express MHC2-associated antigen-derived peptides. Creates the ideal conditions for the onset of an immune response. B. Lymphocytes can interact with membrane-bound antigen while helper T cells recognize MHC2 with antigen-derived peptides presented by the same APC. The N-recognizing cells will interact and co-stimulate each other, and, after a time lag necessary for proliferation and differentiation of B cells into antibody-producing cells, circulating antibody will become detectable. 1. A primary immune response will take 5 to 7 days, sometimes as long as 2 to 3 weeks, to be detected. The predominating isotype of the antibodies made early in a primary immune response is IgM. Institute for Advanced Studies, Jodhpur 76. 2. A secondary immune response has a shorter lag phase, as short as 3 to 4 days, and the predominant isotype of the antibodies is Ig. 3. Differentiation between IgM and Ig antibodies is a useful index for discriminating between recent and past infections, particularly in the case of viral infections. B. Antibody-dependent anti-infectious effector. Mechanisms. As specific antibodies become available, they can protect the organism against infection by several different mechanisms. 1. Complement-mediated lysis of a microorganism or virus-infected cell can result from the activation of the complete sequence of complement. However, as discussed below, both mammalian cells and most pathogenic microorganisms have developed mechanisms that allow them to resist complement-mediated lysis. 2. Opsonization and phagocytosis are, in contrast, of extreme physiological significance. Several proteins can opsonize and promote phagocytosis, as discussed above, but Ig antibodies are the most efficient opsonins, particularly when complement is activated and C3b joins Ig on the microbial cell membrane. A. Opsonins. Because of their role in promoting opsonization, Ig antibodies and C3b are considered opsonins. Both C3b and Ig alone are efficient opsonins, but their effects are synergistic. B. Fc and DR1 receptors, expressed on the membranes of all phagocytic cells, mediate phagocytosis and also deliver activating signals to the cells, inducing release of cytokines, as mentioned above. C. Killing has been demonstrated for bacteria, fungi, and viruses, while phagocytosis of antibody complement-coated unicellular parasites has not been clearly demonstrated. D. The biological significance of phagocytic cells as ultimate mediators of the effects of opsonizing antibodies is obvious. The protective effects of antibodies are lost in patients with severe neutropenia or with severe functional defects of their phagocytic cells, and those patients have increased incidence of infections with a variety of opportunistic organisms. 3. Antibody-dependent cell-mediated cytotoxicity. ADCC. Cells with Fc receptors may be able to participate in killing reactions that target antibody-coated cells. Ig1, Ig3, and IgE antibodies in cells with Fc gamma R or Fc epsilon R are usually involved. Granular lymphocytes or monocytes are the most common effector cells in ADCC, but in the case of parasitic infections, eosinophils play the principal role in cytotoxic reactions. Different effector mechanisms responsible for killing by different types of cells. Large granular lymphocytes kill through the release of granzymes and signaling for apoptosis. Monocytes kill by releasing oxygen-active radicals and nitric oxide. Eosinophil killing is mostly mediated by the release of a major basic protein, which is toxic for parasites. 4. Toxin neutralization. Many bacteria release toxins, which are often the major virulence factors responsible for severe clinical symptoms. Antibodies to these toxins prevent their binding to cellular receptors and promote their elimination by phagocytosis. 5. Virus neutralization. Most viruses spread from an initial focus of infection to a target tissue via the bloodstream. Antibodies binding to the circulating virus change its external configuration and prevent either its binding to cell receptors or its ability to release nucleic acid into the cell. 6. Mucosal protection. Secretory antibodies seem to play their protective role by preventing the attachment and penetration of microbial agents through mucosal surfaces. C. Factors influencing the effectiveness of an anti-infectious humoral response. The effect of the humoral immune response in eliminating or preventing the proliferation of an infectious agent or in neutralizing a toxin before it causes severe disease depends on whether or not antibodies can be available in time to prevent the infection to develop or the toxin to reach its cellular target. 1. Are present in circulation. As a result of vaccination, previous infection, or cross-reaction between different microorganisms, protection is most effective. The microorganism or its toxins will be almost immediately neutralized, and the infection will remain subclinical. 2. If preformed antibodies are not available, protection will depend on whether antibody synthesis can take place before the incubation period of time during which the infectious agent is multiplying but has not yet reached sufficient mass to cause clinical disease, is over. A. Infectious agents, such as the influenza virus, have very short incubation periods, about 2 to 3 days, and, in such cases, not even a secondary immune response can be protective. B. Most infections, the duration of the incubation period is sufficiently long as to allow a secondary immune response to provide protective antibodies. Thus, for many, particularly the common viral diseases of childhood, previous exposure and acquisition of memory ensure that antibody will be produced in time to maintain subsequent exposures, which play the role of natural booster doses, probably explaining the immunity for life associated with them. C. The goal of prophylactic immunization may vary from case to case. In diseases with very short incubation periods, it is essential to maintain the levels of neutralizing antibody in circulation necessary to immediately abort infection. In most rare diseases, it may be sufficient to induce immunological memory, since once memory has been induced, the immune system will be able to respond in time to prevent the development of clinical infection. D. Protect by humoral immunity is only possible if the infectious agent is easily available to the antibodies produced against it. Thus, organisms that grow intracellularly are not easy to eliminate by antibodies. In addition, as discussed in detail later in this chapter, those organisms able to change their antigenic makeup during the course of an infection can persist in spite of a vigorous humoral response. V. The protective role of cell-mediated immunity. Many organisms have the ability to grow and replicate intracellularly, some as an absolute requirement, others as an option that allows them to survive after phagocytosis. Largely ineffective against those organisms, and T lymphocytes play a major role in their elimination, as reflected by the finding of lymphocytic infiltrates in tissues infected by intracellular infectious agents, such as viruses. The immune system. Institute for Advanced Studies, Jodhpur 77. Two basic options to eliminate those organisms, to kill the infected cell, or to enhance the infected cell's ability to kill intracellular organisms. Either option requires the persistent activation of the Th1 subpopulation of helper cells. A. Lymphocyte-mediated cytotoxicity. It can be demonstrated that viral infected cells are lysed as a consequence of their incubation with immune lymphocytes obtained from an animal previously exposed to the same virus. 1. Recognition of infected cells. A. Virus-infected cells that express MHCI molecules with associated viral peptides can be easily recognized by CD8+ cytotoxic T cells. While CD8's lymphocytes, when strongly stimulated, are able to release IL-2 and differentiate into effector cytotoxic T cells, usually the differentiation requires the collaboration of Th1 helper T cells. The CD4+ THO cells and their differentiation into Th1 helper T cells is likely to take place in lymphoid tissues, since macrophages are often infected by viruses and virus-derived peptides are likely to be presented by those cells both in association with MHCI and MHC2 molecules. B. Macrophages are often infected by intracellular organisms of all kinds, including bacteria, fungi, and protozoa. Recognition by CD4+ and CD8+ T lymphocytes is likely to take place, resulting in the differentiation of effector CD8+ cytotoxic T cells. 2. Involves recognition of the target cell, release of perforins and esterases, and delivery of apoptosis signals through the FAS-FAS ligand interaction. B. Lymphocyte-mediated activation of macrophages and other inflammatory cells. In many cases, infected by intracellular organisms of the tissue macrophages, and the persistence of the infection depends on a delicate balance between a state of relative inactivity by the macrophage and mechanisms that allow the infectious agent to escape proteolytic digestion once inside the cytoplasm. See below. 1. Macrophage activation. A. 4+ Th1 lymphocytes are activated as a consequence of their interaction with infected macrophages expressing MHC2-associated peptides and releasing IL-12. Once activated, lymphocytes release a variety of lymphokines, particularly interferon, which activates macrophages, enhancing their ability to kill intracellular organisms, and GM-CSF, which promotes different activated Th lymphocytes release a variety of lymphokines, particularly interferon, which activates macrophages, enhancing their ability to kill intracellular organisms, and GM-CSF, which promotes differentiation and release of granulocytes and monocytes from the bone marrow and delivers costimulatory signals to B cells. B. Once facial activation signals are delivered, macrophages and lymphocytes enter in a complex cycle of self and mutual activation involving a variety of cytokines. In addition, several of these cytokines may activate other types of cells and have chemotactic properties, for which reason they are known as chemotactic cytokines or chemokines. The list of cytokines includes interleukin 8, IL-8, RANS, macrophage inflammatory proteins, MIP, macrophage chemotactic proteins, MCP, and migration inhibition factor, MIF. Collectively, the chemokines attract, activate, and retain leukocytes to the area where a cell-mediated immune reaction is taking place. 2. Inflammatory reaction. In concert with the release of chemotactic cytokines, the expression of CAMs in neighboring microvessels is upregulated, favoring adherence and migration of monocytes and granulocytes to the extravascular space. Inflammatory cells accumulate in the area of infections and, as a consequence of cross-activation circuits involving phagocytes and Th1 lymphocytes, the localized macrophages and granulocytes become activated. Consequently, A. The enzymatic contents and respiratory burst of the cells become more intense, making them better suited for killing intracellular organisms. B. Phagocytosis is enhanced. C. Activated macrophages higher levels of cytokines, including IL-12, which continues to promote the differentiation of Th1 cells, as well as IL-1, IL-6, and TNF-alpha, which in association with IL-8, play the major role in inducing the synthesis of reactive proteins, increasing body temperature, and several other metabolic effects characteristic of the inflammatory reaction. B. The immune deficiency syndromes as models for the study of the importance of immune defenses against infections in humans. Most of our information about the immune system in humans has been learned from a study of patients with immunodeficiency diseases. The most characteristic clinical features of immunocompromised patients are the repeated or chronic infections, often caused by opportunistic agents. Characteristic associations between specific types of infections and generic types of immune deficiency, which provide the best data about the role of the different components of the immune system. A. Patients with antibody deficiencies and conserved cell-mediated immunity suffer from repeated and chronic infections with pyogenic bacteria. B. Patients with primary deficiencies of cell-mediated immunity usually suffer from chronic or recurrent fungal, parasitic, and viral infections. C. Neutrophil deficiencies are usually associated with bacterial and fungal infections caused by common organisms of low virulence, usually kept in check through non-immune phagocytosis and other innate resistance mechanisms. Highlighted complement component deficiencies are also associated with bacterial infections, most frequently involving Neisseria gonorrhoeae and meningitis, whose elimination appears to require the activation of the membrane attack complex. 7. Escape from the immune response. Many infectious agents have developed the capacity to avoid the immune response. Several mechanisms are involved. A. Anti-complementary activity has been characterized for bacterial capsules and outer proteins of some bacteria. For example, the M protein of group of Streptococcus activates complement convertases, preventing activation of the alternative pathway. The anti-complementary activity of bacterial outer components has as a net result a decreased level of opsonization by C3b and other complement fragments. Institute for Advanced Studies, Jodhpur 78. E. Resistance to phagocytosis can be evident at different levels of the sequence of events associated with ingestion and digestion of microorganisms. 1. Resistance to ingestion is usually associated with polysaccharide capsules, which repeal and inhibit the function of phagocytic cells. E. phylis, Pasteurella, Klebsiella, Pseudomonas, and some yeasts, e.g., Cryptococcus neoformans, have such capsules. 2. Ability to survive digestion is characteristic of the group of bacteria known as facultative intracellular mycobacteria, Brucella, Listeria, and Salmonella, as well as of some fungi and protozoa Toxoplasma, Trypanosoma cruzi, and Leishmania. These microorganisms survive inside phagocytosis by several different strategies. A. Of molecule prevent the formation of phagolysosomes. These bacteria survive inside phagosomes that are relatively devoid of toxic compounds, Mycobacterium tuberculosis, Legionella pneumophila, and Toxoplasma gondii. B. Synthesis of outer coats that protect the bacteria against proteolytic enzymes and free toxic radicals, such as the superoxide radical. C. Dependent of the response of the infected phagocytic cells to cytokines which usually activate their killing functions, such as interferon gamma. D. Exit from the phagosome into the cytoplasm, where the bacteria can live and multiply unharmed, Trypanosoma cruzi. E. The resistance to phagocytosis of several organisms seems to involve a combination of these strategies. I. Mycobacterium leprae is coated with a phenolic glycolipid layer that scavenges free radicals and releases a compound that inhibits the effects of interferon gamma. In addition, the release of IL-4 and IL-10 is enhanced, contributing to the downregulation of Th1 lymphocytes. E. Lipids present in the membrane of Leishmania parasites inhibit the response of monocytes and neutrophils to cytokines and a variety of stimuli. In addition, Leishmania seems to resist the effects of proteolytic enzymes in the phagolysosome. F. Viruses taken up by phagocytes as antigen-antibody complexes often infect the phagocytic cells. The infection of phagocytic cells is frequent when viruses disseminate through the bloodstream. G. Phagocytic cells are also infected by the HIV virus, either as a consequence of ingestion of virus-ig antibody complexes or as a consequence of interactions with a CD4+ related molecule which is expressed in their membrane. In the infection of the monocyte, macrophage is not cytotoxic, but the virus continues to replicate in small amounts and these cells become permanent sources of reinfection. C. Ineffective immune responses. 1. Polysaccharide capsules are not only antiphagocytic, but poorly immunogenic and elicit predominantly IgM and Ig2 antibodies, which are inefficient as opsonins. The Fc gamma R of phagocytic cells recognize preferentially Ig1 and Ig3 antibodies. 2. Neisseria meningitidis often induces the synthesis of IgA antibodies. In vitro data that IgA can act as a weak opsonin or induce ADCC, monocytes, macrophages and other leukocytes express Fc alpha receptors on their membranes, but the physiological protective role of IgA antibodies is questionable. Patient sera with high titers of IgA antibodies to N meningitidis failed to show bactericidal activity until IgA-specific anti-Neisseria meningitidis antibodies were removed. This observation suggests that the IgA antibodies acted as a blocking factor, preventing opsonizing IgA antibodies from binding to the same epitopes. 3. Antiviral antibodies are not always neutralizing and protective. A. Retroviruses induce both neutralizing and non-neutralizing blocking antibodies. Both seem to recognize the same epitopes. When non-neutralizing antibodies predominate, they block the binding of neutralizing antibodies. B. Hepatitis B virus induces the synthesis of large quantities of soluble antigenic proteins by the infected host cells. Circulating antigens will block the antiviral antibodies before they reach the infected cells, in a sense, acting as a deflector shield that protects the infected tissues from antibody aggression. D-loss and masking of antigens have been demonstrated with schistosomal, the larval forms of schistosoma. 1. In the few after hatching, there is a rapid decrease in the surface antigens, as reflected by a progressive decrease in the binding of purified and fluorescein-labeled heterologous antischistosomal antibodies to the parasite membrane. This is observed even in parasites cultured in serum-free medium. 2. Schistosoma recovered from infected animals have host proteins bound to the outer layers, masking the remaining parasite antigens. E. Variation has been characterized in bacteria, Borrelia recurrentis parasites, trypanosomes, the agents of African sleeping sickness, Giardia lamblia, and viruses, human immunodeficiency virus, HIV. 1. African somes have a surface coat constituted mainly of a single glycoprotein, variant-specific surface glycoprotein or VSG, for which there are about 103 genes in the chromosome. At any given time, only one of those genes is expressed, the others remain silent. For every 106 or 107 trypanosome divisions, a mutation occurs that replaces the active VSG gene on the expression site by a previously silent VSG gene. The previously expressed gene is destroyed, and a new VSG protein is coded, which is antigenically different. The emergence of a new antigenic code allows the parasite to multiply unchecked. As antibodies reach to the newly expressed VSG protein, parasitemia will decline, only to increase as soon as a new mutation occurs and a different VSG protein is synthesized. 2. Lamblia has a similar mechanism of variation, but the rate of surface antigen replacement is even faster, once every 103 divisions. 3. Borrelia recurrentis, the agent of relapsing fever, carries genes for at least 26 different variable major proteins, VMP, which are sequentially activated by duplicative transposition to an expression site. The successive waves of bacteremia and fever correspond to the emergence of new mutants which, for a while, can proliferate unchecked until antibodies are formed. 4. HIV has a high degree of antigenic variation, which seems to be the result of errors introduced by the reverse transcriptase when synthesizing viral DNA from the RNA template. The mutation rate is relatively high, 1 in every 103 progeny particles, and the Immune Institute for Advanced Studies, Jodhpur 79. Selects the mutant strains that present new configurations in the outer envelope proteins, allowing the mutant to proliferate unchecked by pre-existing neutralizing antibodies. F. Cell-to-cell spread allows infectious agents to propagate without being exposed to specific antibodies or phagocytic cells. 1. Listeria methogenes, after becoming intracellular, can travel along the cytoskeleton and promote the fusion of the membrane of an infected cell with the membrane of a neighboring non-infected cell, which is subsequently invaded. 2. Herpes viruses, retroviruses, and paramyxoviruses cause the fusion of infected cells with non-infected cells, allowing viral particles to pass from cell to cell. Integration of microbial genomes is a tactic exclusive of viruses, particularly DNA viruses and retroviruses. Integration of viral genomes in the host genome allows the virus to cause persistent or latent infections, with minimal replication of the integrated virus on minimal expression of viral proteins in the membranes of infected cells. H. Immunosuppressive effects of infection. Immunosuppressive effects have been described in association with bacteria and parasitic infections. The best documented examples of infection-associated immunosuppression are those described in viral infections. 1. Measles. Patients in the acute phase of measles are more susceptible to bacterial infections, such as pneumonia. Both sensitivity responses and then in vitro lymphocyte proliferation in response to mitogens and antigens are significantly depressed during the acute phase of measles in the immediate convalescence period, usually returning to normal after 4 weeks. Recent investigations suggest that infection of monocytes, macrophages with the measles virus is associated with a downregulation of interleukin-12 synthesis, which can explain the depression of cell-mediated immunity associated with measles. 2. Cytomegalovirus. Mothers infected with cytomegalovirus show depressed responses to CMV virus, but normal responses to T-cell mitogens, suggesting that, in some cases, the immunosuppression may be antigen-specific, while in measles it is obviously nonspecific. 3. Influenza virus has been found to depress CMI in mice, apparently due to an increase in the suppressor activity of T lymphocytes. 4. Epstein-Barr virus releases a specific protein that has extensive sequence homology with interleukin-10. The biological properties of this viral protein are also analogous to those of interleukin-10. Both are able to inhibit lymphokine synthesis by T-cell clones. 5. Human immunodeficiency virus. HIV infection is associated with depletion of CD4+ cells, which are the primary target of the virus. Depletion of CD4 cells results mostly from viral replication itself and by the priming of infected cells for apoptosis, which is triggered by stimuli that normally would cause T-cell proliferation. In addition, FcR-dependent phagocytosis is suppressed in HIV-infected monocyte-derived macrophages, further contributing to the immunological compromise of the infected patients. X. Abnormal consequences of the immune response. A. The activation of T lymphocytes by bacterial superantigens. 1. A variety of bacterial, such as staphylococcal enterotoxins A and B, SEA and SEB, staphylococcal toxic shock syndrome toxin 1, TSST1, exfoliating toxin, and streptococcal exotoxin A, as well as other unrelated bacterial proteins, such as streptococcal M proteins, have been characterized as superantigens. 2. Superantigens are defined by their ability to stimulate T cells without being processed. Stimulation of T cells is polyclonal, thus the designation of superantigen is a misnomer, but it has gained popularity and is widely used in the literature. 3. Studied superantigens are the staphylococcal enterotoxins, which are potent polyclonal activators of murine and human T lymphocytes, inducing T cell proliferation and cytokine release. TSST1 also appears to activate monocytes and is a potent B cell mitogen, inducing B cell proliferation and differentiation. 4. Mechanism of action. Bind directly and simultaneously to the non-polymorphic area of class II MHC on professional accessory cells, macrophages and related cells, and to the B beta chain of the alpha-beta TCR Fig. A. For example, staphylococcal enterotoxins bind exclusively to specific subfamilies of B beta chains that are expressed only by certain individuals. Beta chain regions can be found on 2-20% of a positive individual's T cells, and the cross-linking of the TCR2 and of the APC by the enterotoxin activates all T cells, both CD4 and CD8+, expressing the specific B beta region recognized by the enterotoxin. B. T cell activation induced by superantigens results in the release of large amounts of IL-2, interferon gamma, lymphotoxin TNF-beta, and TNF-alpha. C. Patients infected by bacteria release large amounts of superantigens, e.g., S. aureus-releasing enterotoxins or TSST1 and group A streptococcus releasing, may develop septic shock as a consequence of the systemic effects of these cytokines, which include fever, endothelial damage, profound hypotension, disseminated intravascular coagulation, multi-organ failure, and death. D. After the initial burst of cytokine release, the stimulated T cells either undergo apoptosis or become anergic. This effect could severely disturb the ability of the immune system to adequately respond to superantigen-releasing dysuria. Institute for Advanced Studies, Jodhpur 80. B. Infection is a consequence of the uptake of antigen-antibody complexes. The immune response, in some cases, facilitates the access of infectious agents to cells in which they will be able to proliferate. 1. Macrophages are often infected by intracellular organisms that are ingested as a consequence of opsonization. 2. Escherichia rothani, a bovine enteroparasitic parasite, penetrates the host cells after it has bound complement, particularly C3. Absorption of C3b containing circulating antigen-antibody complexes to the CR1 expressed by red cells allows the parasite to gain access to the red cell, which becomes its permanent location. C. Post-infectious tissue damage. Several examples of deleterious consequences of the immune response have to do with the predominant role of the immune response in the pathogenesis of the disease. 1. Immune complex-induced inflammation. Antigen antibody complexes, if formed in large amounts, can cause disease by being trapped in different capillary networks and leading to inflammation. The clinical immune complex-related inflammation depends on the localization of the trapped complexes. Vasculitis and purpura, when the skin is predominantly affected. Glomerulonephritis if trapping takes place on the glomerular capillaries. Arthritis when the joints are affected. Viruses are often involved in the formation of circulating antigen-antibody complexes. 2. Immune destruction of infected cells and tissues. An immune response directed against an infectious agent may be the main cause of damage to the infected tissue. A. In subacute sclerosing panencephalitis, a degenerative disease of the nervous system associated with persistent infection with the measles virus, the response against viral epitopes expressed in infected neurons is believed to be the primary mechanism of disease. B. Forms of chronic active hepatitis, the immune response directed against viral epitopes expressed by infected hepatocytes seems to cause more tissue damage than the infection itself. C. Cross-reactions with tissue antigens have been proposed as the basis for the association of streptococcal infections with rheumatic carditis and glomerulonephritis. Antibodies to type 1 streptococcal M protein cross-react with epitopes of myocardium and kidney mesangial cells and cause inflammatory changes in the heart and glomeruli, respectively. X. Epilogue. The outcome of an infectious process depends on a very complex set of interactions with the immune system. A successful pathogen has developed mechanisms that avoid fast elimination by an immunocompetent host. These mechanisms allow the infectious agent to replicate, cause disease, and spread to other individuals before the immune response is induced. The immune response, on the other hand, is a powerful weapon that, once set in motion, may destroy friendly targets. Thus, therapeutic strategies in infectious diseases have to consider all these questions, such as the particular survival strategy of the infectious agents, the effects of the infection on the immune system, and the possibility that the immune response may be more of a problem than the infection itself. Unit 5. Institute for Advanced Studies, Jodhpur 1. A. Basic concepts of development. The processes of development. Development is a process in which an organism undergoes a series of progressive changes, taking on the successive forms that characterize its life cycle. Figure 1. In its earliest stages of development, a plant or animal is called an embryo. Sometimes the embryo is contained within a protective structure, such as a seed coat, an eggshell, or a uterus. Embryo does not photosynthesize or feed actively. Instead, it obtains its food from its mother directly or indirectly by way of nutrients stored in a seed or egg. A series of embryonic stages may precede the birth of the new, independent organism. Most organisms continue to develop throughout their life cycle. Development ceases only with death. Development consists of growth, differentiation, and morphogenesis. Three processes are responsible for the developmental changes an organism undergoes during its life cycle. Growth, increase in size, occurs through cell division and cell expansion. In all multicellular organisms, repeated meiotic divisions generate the multicellular body. In plants, cell expansion begins shortly after the first divisions of the fertilized egg. In animals, on the other hand, cell expansion is often slow to begin. The animal embryo may consist of thousands of cells before it becomes larger than the original fertilized egg. Growth continues throughout the individual's life in some species, but reaches a more or less stable endpoint in others. Differentiation is the generation of cellular specializations. That is, differentiation defines the specific structure and function of a cell. Mitosis produces daughter nuclei that are chromosomally and genetically identical to the nucleus that divides to produce them. However, the cells of a multicellular organism are obviously not all identical in structure or function. This apparent contradiction results from regulation of the expression of various parts of the genome. When the embryo consists of only a few cells, each cell has the potential to develop in many different ways. Development proceeds, however, the possibilities available to individual cells gradually narrow, until each cell's fate is fully determined and the cell has differentiated. Morphogenesis, literally, creation of form, is the shaping of the multicellular body and its organs. Morphogenesis results from pattern formation, the organization of differentiated tissues into specific structures. In plant development, cells are constrained by cell walls and do not move around the body, so organized division and expansion of cells are the major processes that build the plant body. In animals, cell movements are very important in morphogenesis. And in both plants and animals, programmed cell death is essential to orderly development. Like differentiation, morphogenesis results ultimately from the regulated activities of genes and their products, as well as from the interplay of extracellular signals and their transduction in target cells. Institute for Advanced Studies, Jodhpur 2. As development proceeds, cells become more and more specialized. Experiments in which specific cells of an early embryo were marked with stains have revealed which adult structures are derived from which parts of the embryo. These stained embryos produce what are known as fate maps. For instance, we know that the green shaded area of the frog embryo shown in Figure 2 normally becomes part of the skin of the tadpole larva. However, if we cut out a piece from this region and transplant it to another location on another early frog embryo, it does not become skin. The type of tissue it does become is determined by its new environment. The developmental potential of these early embryonic cells, that is, their range of possible fates, is thus greater than their actual fate, which is limited to the cell type that normally develops. Does embryonic tissue retain its broad developmental potential? Generally speaking, the answer is no. The developmental potential of cells becomes restricted fairly early in normal development. Tissue from a later stage frog embryo, for example, if taken from a region fated to develop into the brain, becomes brain tissue even if transplanted to part of an early stage embryo destined to become another structure. The cells of the later stage embryo are thus said to be determined. Their fate has been sealed, regardless of their surroundings. By contrast, the cells of the younger tissue transplant in Figure 2 have not yet become determined. Determination, the commitment of a cell to a particular fate, is a process influenced by the action of the extracellular function. This apparent contradiction results from regulation of the expression of various parts of the genome. When the embryo consists of only a few cells, each cell has the potential to develop in many different ways. As development proceeds, however, the possibilities available to individual cells gradually narrow, until each cell's fate is fully determined and the cell has differentiated. Morphogenesis, literally, creation of form, is the shaping of the multicellular body and its organs. Morphogenesis results from pattern formation, the organization of differentiated tissues into specific structures. Plant development, cells are constrained by cell walls and do not move around the body, so organized division and expansion of cells are the major processes that build the plant body. In animals, cell movements are very important in morphogenesis, and in both plants and animals, programmed cell death is essential to orderly development. Like differentiation, morphogenesis results ultimately from the regulated activities of genes and their products, as well as from the interplay of extracellular signals and their transduction in target cells. As development proceeds, cells become more and more specialized. Experiments in which specific cells of an early embryo were marked with stains have revealed which adult structures are derived from which parts of the embryo. These stained embryos produce what are known as fate maps. For instance, we know that the green shaded area of the frog embryo shown in Figure 2 normally becomes part of the skin of the tadpole larva. However, if we cut out a piece from this region and transplant it to another location on another early frog embryo, it does not become skin. The type of tissue it does become is determined by its new environment. Developmental potential of these early embryonic cells, that is, their range of possible fates, is thus greater than their actual fate, which is limited to the cell type that normally develops. Does embryonic tissue retain its broad developmental potential? Generally speaking, the answer is no. The developmental potential of cells becomes restricted fairly early in normal development. Tissue from a later stage frog embryo, for example, if taken from a region fated to develop into the brain, becomes brain tissue even if. Institute for Advanced Studies, Jodhpur 3. Transplanted to part of an early stage embryo destined to become another structure. The cells of the later stage embryo are thus said to be determined. Their fate has been sealed, regardless of their surroundings. By contrast, the cells of the younger tissue transplant in Figure 2 have not yet become determined. Determination, the commitment of a cell to a particular fate, is a process influenced by the action of the extracellular environment and the contents of the cell on the cell's genome. Determination is not something that is visible under the microscope. Cells do not change their appearance when they become determined. Determination is followed by differentiation, the actual changes in biochemistry, structure, and function that result in cells of different types. Differentiation often involves a change in appearance as well as function. Determination is a commitment. The final realization of that commitment is differentiation. The role of differential gene expression in cell differentiation. Differentiated cells are recognizably different from one another, sometimes visually as well as in their protein products. For example, certain cells in our hair follicles continuously produce keratin, the protein that makes up hair, nails, feathers, and porcupine quill. Other cell types in the body do not produce keratin. In our follicle cells, the keratin encoding gene is transcribed. In most other cells in the body, that gene is not transcribed. Activation of the keratin encoding gene is a key step in the differentiation of hair follicle cells. From examples like this one, we may say that differentiation results from differential gene expression, that is, from the differential regulation of transcription, post-transcriptional events such as mRNA splicing, and translation in different cell types. Because the fertilized egg, or zygote, has the ability to give rise to every type of cell in the adult body, we say it is totipotent. Its genome contains instructions for all of the structures and functions that will arise throughout the life cycle. Later in the development of animals, the cellular descendants of the zygote lose their totipotency and become determined. These determined cells then differentiate into specific types of specialized cells. With differentiation, there is generally no irreversible change in the genome. Differentiation is irreversible in certain types of cells. Examples include the mammalian red blood cell, which loses its nucleus during development, and the trachid, a water-conducting cell found in many plants. Trachid development culminates in the death of the cell, leaving only the pitted cell walls that formed while the cell was alive. In both of these extreme cases, the irreversibility of differentiation can be explained by the absence of a nucleus. Generalizing about the reversibility of differentiation in mature cells that retain functional nuclei is more difficult. We tend to think of plant differentiation as reversible and of animal differentiation as irreversible, but this is not a hard and fast rule. Whilst differentiation apparently reversible in some cases, such as a plant cutting, but not in others, such as a mammalian limb. At some stage of development, do changes within the nucleus permanently commit a cell to specialization? For both plants and animals, the answer appears to be no. Under the right environmental circumstances, differentiation is reversible in many cells. TOTIPOTENCY in plants, a food storage cell in a carrot root normally faces a dark future. It cannot photosynthesize or give rise to new carrot plants. However, if we isolate that cell from the root, maintain it in a suitable nutrient medium, and provide it with appropriate chemical cues, we can fool the cell into acting as if it were a fertilized egg. It can divide and give rise to a mass of indifferentiated cells, called a callus, and eventually to a complete plant. Figure 3. Since a new plant is genetically identical to the somatic cell from which it came, we call the plant a clone. Institute for Advanced Scientists to clone an entire carrot plant from a differentiated root cell indicates that the cell contains the entire carrot genome and that it can express the appropriate genes in the right sequence. Many types of cells from other plant species show similar behavior in the laboratory. This ability to generate a whole plant from a single cell has been invaluable in agricultural biotechnology. TOTIPOTENCY in early embryonic animal cells. Experiments with plants have established that somatic cells are totipotent. A more direct demonstration that all the genetic material is present in somatic cells has come from nuclear transplantation experiments. These experiments were first done on frogs by Robert Briggs and Thomas King, who asked whether the nuclei of early frog embryos have lost the ability to do what the totipotent zygote nucleus could do. They first removed the nucleus from an unfertilized egg, thus forming an enucleated egg. With a very fine glass tube, they punctured a cell from an early embryo and drew up part of its contents, including the nucleus, which they injected into the enucleated egg. They stimulated the eggs to divide, and many went on to form embryos, tadpoles, and eventually, frogs. These experiments led to two important conclusions. No information is lost from the nuclei of cells as they pass through the early stages of embryonic development. This fundamental principle of developmental biology is known as genomic equivalence. The cytoplasmic environment around a nucleus can modify its fate. Similar experiments have been performed on rhesus monkeys, in which a single cell can be removed from an 8-cell embryo and fused with an enucleated egg. This cell fusion technique causes the nucleus of the embryonic cell to enter the egg cytoplasm. The resulting cell acts like a zygote, forming an embryo, which can be implanted into a foster mother, who ultimately gives birth to a normal monkey. The remaining 7 cells from the original embryo can similarly give rise to offspring by the same cell fusion technique. In humans, the totipotency of early embryonic cells permits both genetic screening and in vitro fertilization. An 8-cell human embryo can be isolated in the laboratory and a single cell removed and examined to determine whether a harmful genetic condition is present. Each remaining cell, being totipotent, can be stimulated to divide and form an embryo, which can be implanted into the mother's uterus, where it develops into an infant. TOTIPOTENCY in adult somatic cells. Cell cloning of animals was very difficult until the late 1990s, when Ian Wilman and his colleagues at a biotechnology company in Scotland used the cell fusion procedure to clone sheep. Figure 4. Previous attempts to produce mammals by this method had worked, as in the rhesus monkey case, only if the donor nucleus was from an early embryo. Apparently, the mammalian donor cells were in the G2 phase of the cell cycle and were fused with the cytoplasm of eggs that were also in G2. Some extradinner replication took place that created havoc with the cell cycle in the egg when it attempted to divide. Wilmot took differentiated cells from a ewe's udder and starved them of nutrients for a week, thus halting the cells in G1 phase of the cell cycle. One of these cells was fused with an enucleated egg from a different breed of ewe. When meiotic inserts in the egg cytoplasm were stimulated, the donor nucleus entered S phase, and the rest of the cell cycle proceeded normally. After several cell divisions, the resulting early embryo was transplanted into the womb of a surrogate mother. Out of 277 successful attempts to fuse adult cells with Institute for Advanced Studies, Jodhpur 5. Enucleated eggs, one lamb, named Dolly, survived to be born. DNA analyses confirmed that Dolly was genetically identical to the ewe from whose udder the donor nucleus had been obtained. A major goal of Wilmot's experiment was to develop a method of cloning sheep that would produce products such as pharmaceuticals in their milk. Cloning procedure could make multiple, identical copies of transgenic sheep that are reliable producers of drugs such as alpha-1 antitrypsin, which is used to treat people with emphysema or cystic fibrosis. The trick of starving donor cells for cloning has been applied to other mammals. Mice have been cloned using the somatic cells surrounding the egg as a source of donor nuclei. Figure 5. Cattle have been cloned to preserve a rare breed in New Zealand. Genetically engineered goats have been cloned to produce several useful proteins in their milk. And, as we described at the beginning of this chapter, cloning is being done to preserve and expand endangered species. A private company has been set up that will clone your pet by nuclear transfer. This flurry of cloning has touched off a flurry of controversy, but cloning is not a new scientific concept. The idea of totipotency was accepted long before Dolly was born, but achieving it is an impressive technical achievement. An example of nuclear totipotency gonorrhea occurs in a human tumor called a teratocarcinoma. Here, a differentiated cell dedifferentiates to form an unspecialized, then it divides, forming a tumor, as occurs in most cancers. But some cells in the tumor redifferentiate to form specialized tissue arrangements. So the tumor can form a large mass of cells inside the abdomen, with some of the cells forming kidney tubules, others hair, and still others teeth. How this redifferentiation occurs is not clear. Stem cells can be induced to differentiate by environmental signals. Genomics implies that a differentiated cell stays specialized because of its environment, not because of its genes, and that appropriate environmental changes could result in a new pattern of differentiation. In normal development, a complex series of signals results in the patterns of differentiation we see in a newborn organism. If these signals could be described in enough detail, we should be able to understand how any cell type becomes any other. In ants, the growing regions at the tips of the roots and stems contain meristems, which are clusters of undifferentiated, rapidly dividing cells. These cells can give rise to the specialized cell types that make up roots and stems, respectively. Plants have many fewer, 15 to 20, cell types than animals, as many as 200. Most plant cell types differ in the structure of their cell walls, whereas most animal cell types have specific cytoplasmic characteristics and many cell-specific proteins. In mammals, stem cells are found in adult tissues that need frequent cell replacement, such as the skin, the inner lining of the intestine, and the blood system. As they divide, stem cells produce cells that differentiate to replace dead cells and maintain tissues. In the body, stem cells have limited abilities to differentiate. Stem cells in bone marrow, for example, produce only the various types of red and white blood cells, while the stem cells in the nervous system produce only the various types of nerve cells. Can one kind of stem cell be manipulated by its environment to produce cells that differentiate into cells of another tissue? The answer appears to be yes. When stem cells from the brain were transplanted into the bone marrow of mice whose bone marrow stem cells had been depleted, they proceeded to act like bone marrow stem cells, producing blood cells. In the reverse experiment, bone marrow stem cells were implanted into the brains of mice, where they formed nerve cells. These experiments indicate that some component of the environment, presumably acting through intercellular signals, determines what a stem cell will do. The stem cell populations that are closest to totipotency are not the ones found in adults, but those of the early embryo. In mice, these embryonic stem cells can be removed from an early embryo, called a blastocyst, and then induced to differentiate in some particular way. Normally, these cells are formed a few days after fertilization, and their fate in the developing embryo is soon determined. Before that time, however, they are virtually totipotent. Such cells can be grown indefinitely in the laboratory and, when injected back into a mouse blastocyst, will mix with the resident cells and differentiate to form all the cell types of the mouse. This kind of experiment shows that blastocyst cells do not lose any of their developmental potential while growing in the laboratory. Embryonic stem cells growing in the laboratory can be induced to differentiate if the right signal is provided. Figure 6. Treatment of mouse embryonic stem cells with a derivative of vitamin A causes them to form nerve cells, while other growth factors induce them to form blood cells, again demonstrating their developmental potential in the roles of environmental signals. This finding raises the possibility of using stem cell cultures as sources of differentiated cells for clinical medicine. A key advance toward this use has been the ability to grow human embryonic stem cells in the laboratory. Institute for Advanced Studies, Job 6. A source for embryonic stem cells could be human embryos made for in vitro fertilization. This medical procedure is used by couples who want a child but cannot conceive naturally. Up to 10 eggs are taken from the mother's ovaries and exposed to the father's sperm, with the hope that some early embryos will form. A few of these embryos are then implanted into the mother's uterus for development. Any remaining embryos not used for implantation could be a source of stem cells. Problem arises if these embryonic stem cells are induced to differentiate to form a tissue for transplantation, say, pancreatic tissue for a patient with diabetes. The cells in the recipient are genetically different, so the recipient's immune system may reject the transplanted cells. This problem has led to the proposal of therapeutic cloning, in which nuclear transplantation and stem cell technologies would be combined. This procedure would require several steps. Eggs are removed from a female donor. An egg is enucleated. A cell is removed from the recipient. The entire cell, or its nucleus only, is fused with the enucleated egg. The egg is stimulated to divide. Embryonic stem cells form. These cells are genetically the recipients. The stem cells are induced to differentiate into the desired tissue for transplantation. Progress with this ambitious program has been slow but steady. The age of custom-made cells to replace those lost to disease or injury is rapidly approaching. Genes are differentially expressed in cell differentiation. Nuclear transplantation, cell fusion, and plant cell cloning have demonstrated genomic equivalence in somatic cells of an organism. Molecular experiments have provided even more convincing evidence. For example, the gene for beta globin, one of the protein components of hemoglobin, is present and expressed in red blood cells as they form in the bone marrow of mammals. Is the same gene also present, but unexpressed, in nerve cells in the brain, which do not make hemoglobin? Nucleic acid hybridization can provide an answer. A probe for the number 1F globin gene can be applied to DNAfrom both brain cells and immature red blood cells. Recall that mature red blood cells lose their nuclei and DNA. In both cases, the probe finds its complement, showing that the beta globin gene is present in both types of cells. On the other hand, if the probe is applied to cellular mRNA, rather than cellular DNA, it finds beta globin mRNA only in the red blood cells, and not in the brain cells. This result shows that the gene is expressed in only one of the two tissues. Many similar experiments have shown convincingly that differentiated cells lose none of the genes that were present in the fertilized egg. What leads to this differential gene expression? One example of differentiation is the conversion of undifferentiated muscle precursor cells, called myoblasts, into the large, multinucleated muscle fibers that make up mammalian skeletal muscles. The key event that starts this conversion is the expression of MyoD1, myoblast determination gene 1. The main product of this gene is a transcription factor, MyoD1, with a helix loop helix domain, which not only binds to the promoters of muscle-determining genes to stimulate their transcription, but also acts on its own promoter to keep its levels high in the myoblasts and in their descendants. Strong evidence for the controlling role of MyoD1 in muscle fiber differentiation comes from experiments in which a sequence containing an active promoter adjacent. Institute for Advanced Studies, Job 7. To MyoD1 is transfected into the precursors of other cell types. For example, if this sequence is added to fat cell precursors, the fat cells are reprogrammed to become muscle cells. Genes such as MyoD1 that direct fundamental decisions in development, often by regulating genes on other chromosomes, usually encode transcription factors. The roles of cytoplasmic segregation and induction in cell determination. What initially stimulates the MyoD1 promoter to begin transcription is not clear, but chemical signals are clearly involved in cell differentiation. In general, two overall mechanisms for producing such signals have been found. Cytoplasmic segregation. A factor within an egg, zygote, or precursor cell is unequally distributed in the cytoplasm. After cell division, the factor ends up in some daughter cells or regions of cells, but not others. Induction. The factor is actively produced and secreted by certain cells to induce other cells to differentiate. Polarity results from cytoplasmic segregation. As we learned from the cloning experiments described above, cell nuclei do not undergo irreversible changes during early development, so we must look for explanations of some embryological events in the cytoplasmic differences between cells. The development of polarity, the difference between one end of an organism and the other, is one such phenomenon. Polarity is obvious throughout development. Our heads are distinct from our feet, and the distal ends of our arms, wrists and fingers differ from the proximal ends, shoulders. An animal's polarity may develop early, even in the egg itself, in which yolk and other factors may be distributed asymmetrically. Experiment with sea urchins demonstrates the effects of cytoplasmic segregation on development. Figure 7. Very early development in this species occurs by equal meiotic divisions of the fertilized egg. There is no increase in size at this stage. If an 8-cell embryo is cut vertically, both halves develop normally. On the other hand, if the embryo is cut horizontally, the top half does not develop at all and the bottom half develops into a small, abnormal embryo. Really, then, there must be at least one factor essential for development that is segregated in the bottom half of the egg, such that the bottom cells of the embryo have it, but the top ones do not. Then many experiments have established that certain materials, called cytoplasmic determinants, are distributed unequally in the egg cytoplasm, and that these materials play a role in directing the embryonic development of many organisms. Figure 8. Tissues direct the development of their neighbors by secreting inducers. Experimental work on developing embryos has clearly established that in many cases, the fates of particular tissues are determined by interactions with other specific tissues in the embryo. In developing animal embryos there are many such instances of induction, in which 1. Institute for Advanced Studies, Jodhpur 8. Tissue causes an adjacent tissue to develop in a particular manner. These effects are mediated by intercellular biochemical communication, that is, by chemical signals and signal transduction mechanisms. We will describe two examples of such induction, one in the developing vertebrate eye, and the other in a developing reproductive structure in the nematode C. elegans. The development of the lens of the vertebrate eye is a classic example of induction. In a frog embryo, the developing forebrain bulges out at both sides to form the optic vesicles, which expand until they come into contact with the cells of the surface of the head. Figure 9. The surface tissue in the region of contact with the optic vesicles thickens, forming a lens placode. The lens placode bends inward, folds over on itself, and ultimately detaches from the surface tissue to produce a structure that will develop into the lens. If the growing optic vesicle is cut away before it contacts the surface cells, no lens forms. Placing an impermeable barrier between the optic vesicle and the surface cells also prevents the lens from forming. These observations suggest that the surface tissue begins to develop into a lens when it receives a signal, an embryonic inducer, from the optic vesicle. The interaction of tissues in eye development is a two-way street. There is a dialogue between the developing optic vesicle and the surface tissue. The optic vesicle induces lens development, and the developing lens determines the size of the optic cup that forms from the optic vesicle. If head surface tissue from a frog species with small eyes is grafted over the optic vesicle of one with large eyes, both lens and optic cup will have an intermediate size. The developing lens also induces the surface tissue over it to develop into a cornea, a specialized layer that allows light to pass through and enter the eye. Thus a chain of inductive interactions participates in the development of the parts required to make an eye. Embryonic inducers trigger a sequence of gene expression in the responding cells. Tissues do not induce themselves, rather, different tissues interact and induce one another. Single cells can induce changes in their neighbors. The T. nematode Caenorhabditis elegans is used as a model organism in many biological studies, but it is especially useful for studying development. It normally lives in the soil, where it feeds on bacteria, but can also grow in the laboratory if supplied with its food source. The process of development from fertilized egg to larva takes only about 8 hours, and the worm reaches the adult stage in just 3.5 days. The process is easily observed using a low magnification dissecting microscope because the body covering is transparent. Figure 10a. For all these reasons, C. elegans is a favorite experimental organism. The field of C. elegans does not vary, so it has been possible to identify the source of each of the 959 somatic cells of the adult form. The adult nematode is hermaphroditic, containing both male and female reproductive organs. It lays eggs through a pore called the vulva on the ventral belly surface. During development, a single cell, called the anchor cell, induces the vulva to form. If the anchor cell is destroyed by laser surgery, no vulva forms. The eggs develop inside the parent, in a bag of worms, which eventually consume the parent. Results. The anchor cell controls the fates of 6 cells on the animal's ventral surface through 2 molecular switches. Each of these cells has 3 possible fates. It may become a primary vulval precursor cell, a secondary vulval precursor cell, or simply part of the worm's surface, an epidermal cell. Figure 10b. The anchor cell produces an inducer that diffuses out of the cell and interacts with adjacent cells. Cells that receive enough of the inducer become vulval precursor cells. Cells slightly farther from the anchor cell become epidermis. The first molecular switch, controlled by the inducer from the anchor cell, determines whether a cell takes the track toward becoming part of the vulva or the track toward becoming epidermis. The cell closest to the anchor cell, having received the most inducer, differentiates into the primary vulval precursor cell. It produces its own inducer, which acts on the 2 neighboring cells and directs them to become secondary vulval precursor cells. Thus, the primary vulval precursor cell controls the second molecular switch, determining whether a vulval precursor will take the primary track or the secondary track. The 2 inducers control the activation or inactivation of specific genes in the responding cells. There is an important lesson to draw from this example. Much of development is controlled by molecular switches that allow a cell to proceed down one of 2 alternative tracks. One challenge for the developmental biologist is to find these molecular switches and determine how they work. The primary inducer released by the C. elegans anchor cell appears to be a growth factor homologous to the mammalian epidermal growth factor EGF. The nematode growth factor, called Win3, binds to a receptor on the surface of a potential vulval precursor cell. This binding sets in motion a signal transduction cascade involving the ROS protein and MAP kinases. The end result is increased transcription of the genes involved in the differentiation of vulval cells. Institute for Advanced Studies, Jodhpur 9. The role of pattern formation in organ development. Pattern formation, the spatial organization of a tissue or organism, is inextricably linked to morphogenesis, the appearance of body form. The differentiation of cells is beginning to be understood in terms of molecular events, but how do molecular events contribute to the organization of multitudes of cells into specific body